Preparing for the multiple choice exam, some interesting questions, part 3, this time from the June 2003 paper. In this question, the graph is looking at the change in the length of some plant tissue depending on the concentration of sucrose solution that they were placed in. The question is asking you to identify which concentration of sucrose has the same water potential as the cell sat before immersion, i.e. which concentration of sucrose solution finds the plant tissue in equilibrium. You can work this out because if the plant tissue is in equilibrium with the solution, there will be no change in the length of the plant tissue. Therefore, initial length divided by final length will be 1. So to calculate this, you draw a line from 1 and then you draw a line down from where that meets the line. Slightly odd way of doing it. And then you read the scale. So 0.45, so C is our answer. Lots of information in this question, and the first thing that I would do is assign a colour to each of the columns. So solution 1 is green, solution 2 is blue, solution 3 is red, and solution 4 is yellow. Okay, and that just appeals to my sense of visual learning. Then you need to think, well, it's asking you basically to identify which solution has the lowest through to the highest concentration. So this one should be the lowest, this one should be the highest, okay? So it should go lowest 1, 2, 3, 4, because the change in colours are showing the change in the concentration of the reducing sugar, okay? So what are we looking at here? Well, in this column, it could be the reducing sugar, 0.5% reducing sugar, or indeed a 1% reducing sugar, non-reducing sugar, sorry. Either of those would be correct. Okay, so we're now looking at line A or line C. Okay, we can discount line B and line D because if there was any reducing sugar in the solution, you would get a change in colour. So then you need to identify, for the rest of this, which is the better line, A or C. Well, in line C, it's saying that this column has a higher concentration than this column, which is nonsense, so it must be line A. This is another favourite experiment. So the easiest way to look at this is to just draw it out. So here is your heavy DNA, okay? So the black lines represent the heavy DNA. After one generation, you're going to have half and half because you're going to have your semi-conservative replication. After 2 generations, you're going to have that plus 2 normal lines, okay, and so on. And so to draw it out and then identify which line is going to be the best line, and you will come up with C as your answer. Here is another question looking at the cardiac cycle graph. This time it's asking you to identify what is happening to the ventricle and the aortic semilunar valve at X. Well, X is the point where the pressure in the ventricle is higher than the pressure in the aorta, so that means that the semilunar valve has just opened. Obviously the pressure is increasing in the ventricle. The only thing that could be causing the pressure to increase is ventricular systole, and therefore answer B, contracting and the valve opening, is correct. I think the best way to answer this question is to draw where the water would move before you worry about what the question is actually asking you to do. So water is always going to move from a high, less negative to a low, more negative water potential. So it's going to move from cell 2 to cell 1, from cell 2 to cell 3, from cell 3 to the environment, from cell 2 to the environment, and from the environment into cell 1. So now that you've identified how the water is going to move, you can then determine which of the 4 statements is correct. So all 3 cells are turgid, so no water moves. Water moves from cell 1, cell 3, and the environment into cell 2. Water moves from cell 3 to the environment, and from the environment to cell 1. Yes. But let's just check that the next one can't be right. Water moves from the environment into cells 1, 2, and 3. No. So the answer is C. This is a good opportunity to use elimination. So you know that number 1 is nitrogen fixing bacteria. So that enables you to cross out lines A and line C straight away. Okay. So then you just need to pick which of 2 and 3 you're most confident in, which one's which. Well, nitrate to nitrogen gas that was the denitrifying bacteria, so number 3, so therefore B is our answer.